patients. How much total follow-up do you have at this point, and can you talk about what trends you're seeing beyond 28 days?
Thanks, Maury. It's John. That's information that we'll share later this year as we collect it. Remember that we are trying to share information on a cohort-by-cohort basis so we get a consistent readout. So as the year goes on, we'll be in a position to share that information when we have a more complete picture.
Got it. Okay. And then in the New England Journal of Medicine publication, it mentions plans for longer-term safety follow-up under a separate program in development. So I'm just wondering when we'll learn more about the separate program, and will this program enable you to redose cohort 1 and potentially get biopsies from any of the patients treated?
Well, thanks, Maury, again. Biopsies are really not necessary nor informative, and so I don't think that's going to be relevant to the program. As we've laid out from the outset, given that this is a single ascending dose phase of the program, for those patients who turn out to have a dose that was less than maximally efficacious, we're making provisions for those patients to come back and get access to that dose, and that would be outside this particular protocol. The details of that are something that we'll share a little bit later on as we get more information. I think just to make sure everybody understands, one of the reasons we're excited about the LNP platform is that, in fact, you can redose. And this was a very important part of putting the modular system together and the chemical-based approach as opposed to a virological delivery. And so as we learn how the lower doses behave, we're able to take the information that we gather at higher doses and make it available to those who came in early in the study. So we should all be in a good position for everybody to benefit from that information as we go.
Great. Okay, thanks for taking my questions. I'll hop back in the queue.
Sure.
The next question comes from Salveen Richter from Goldman Sachs. Please go ahead.
Good morning and congratulations on the data. Just a couple of questions for me. How are you monitoring for potential off-target edits for patients in the study? And if you could comment on the read-through to cardiomyopathy, and then as well, given you have read-through here to HAE, anything you can give us around trial design there or starting dose, given the work you've seen now in TTR polyneuropathy? Thank you.
Thanks, Helene. As we've tried to make clear in other venues, the off-target aspect is something that is substantially de-risked preclinically by extensive work that's done in human cells at suprapharmacologic concentrations. I think Dr. Gilmore tried to address that. What we've showed is that using the most stringent assays that anybody is applying, we test concentrations that are far in excess of what any patient's ever going to get. And what we've demonstrated is that at those concentrations that give the results reported today, there's no interaction with sequences other than the targeted one. So we're quite confident that from the standpoint of off-targets, we know exactly what we're doing. We know exactly where CRISPR is producing its effects. So we think we're in quite good shape there. With respect to cardiomyopathy, the biology is the same. The edit that we get with this particular approach in these polyneuropathic patients is expected to be identical in the cardiomyopathic patients, and we expect to show that here quite soon as we move into that patient population. And maybe I'll turn to David Lebwohl, our CMO. David, if you just want to give some basic ideas of how we're thinking about HAE and how these results relate to that protocol.
Yeah, thanks, John. So the HAE drug, I think is part of the platform. So the only difference from 2001 are the 20 nucleotide sequence at the 5' end. So this is really the same lipid nanoparticle, the same messenger RNA, and almost the same guide. So based on this, we think we know already a good bit about what the safety is expected to be with HAE. And on that basis, we do think we could potentially start at a higher dose, potentially at the second dose, where we've seen only grade 1 safety events in this population, in the TTR population. The HAE population is quite healthy as well, and we do expect them to do very well. So we're not talking about the exact design of the phase 1 yet. I think you'll have some sense just from what you know already, but we will be talking more about that as we get close to the study.
Thank you.
The next question comes from Gina Wong from Barclays. Please go ahead.
Thank you for taking my questions. I wanted to say that this is a transformative for the whole field. I'm really really excited to see this data. Congratulations. So I have 3 sets of questions. First, John, regarding the off-targets, we understand that, I think Dr. Gilmore also mentioned, human cell line is a very good system to test the off-targets, and you did show very good limited off-targets. But from the FDA perspective, do you think that they wanted to see actual data from humans? That's the first question. And the second question is regarding non-human primates data. It seems like translation from non-human primates data to humans seems much better. When we look at, Alnylam actually showed pretty consistent with FDA guidance, basically 3-fold differences, and Alnylam data showed very consistent, but in your case seems much better. So any thoughts on, did you optimize the lipid nanoparticle or the final drug product? Any thoughts there explaining that? And the last question is, beyond for the HAE program, I assume your first dose will be 0.3 milligram per kg, if that's the case. And then also beyond HAE, what other knocking down indication you wanted to go after?
Thank you, Gina. There's a lot there. First of all, the work that we do with off-targets is done in primary hepatocytes, not cell lines. And we think that gives us far and away the best sort of determination and assessment of what's going on. This is information we've shared with the regulators. They've seen how provocative we've done our testing, again, at suprapharmacologic levels, and that's been satisfactory. There's been no request for testing in humans, and I think just about anybody who thinks about it for a while realizes that that's not going to be particularly informative. So I would not look for that type of work as we go down the road here. With respect to non-human primates, well, first of all, we were very pleased with the extent that we were able to predict, or at least come close to what we hoped to achieve in humans. There's lots of precedents. We have a great team that's done a lot of work extrapolating back and forth between different cell types and the preclinical models. We've also learned that the human guide is quite active, and you have to take that into consideration relative to the non-human primate guide, and there's an additional benefit if you have activity there. So, I guess what we can take away from this is that standard allometric thinking is a very good guidepost, which is good news for us because we can take the information that's been built over the years as people been thinking about going from preclinical models to the clinic and benefit from those insights. And then, capture the additional data that's, particular to the test article that we're doing. So we're quite excited. I give my preclinical team a strong A with the work that they've done, and we think that it puts us in a really good position for where we go with HAE. Specifically with the HAE protocol, I wouldn't spend too much time trying to guess what dose we're going to start with. Just as we did with the TTR work, we try to be very thoughtful about the patient population, what we know from preclinical models, obviously what we're learning from this particular study. But, the takeaway, I think, should be that we can start at doses that are higher than the lowest dose here. What that particular dose will be is something that we'll decide as we begin enrolling those patients. So overall, I think that the takeaway is that the preclinical work is solid. It teaches us a lot, and it's been an excellent guide for what we do in the clinic. And that's a very, very good place to be in.
Our next question comes from Mani Foroohar from SVB Leerink. Please go ahead.
Hey guys, congratulations on the data and for publishing in our small local medical journal here in Boston. A couple of quick questions for you on this one. One of the pushbacks that we've gotten is some of the complexity and challenges in terms of clinical development and indication with many therapies available or soon to be available as TTR. The players who approved RNAis have been able to reach into the placebo data they had from previous studies to accelerate and provide synthetic control arms for their next generation products. Are there opportunities for you to look at existing placebo data from other studies as a potential control arm, or would you expect an active control? And then I have a quick follow-up question.
We will look at any database that we have access to. If there's proprietary databases that we can't access, well, that's just the way it is. Obviously, we're going to work with experts like Dr. Gilmore and people who are very, very well versed in the space to design a protocol that builds on the information that's accumulated. And with the particulars of what the control will be, I mean, that's something that we'll think through. We have some ideas already on how to do that in a very, very efficient fashion. And I don't think today's the place to go into what those particular studies will be while we're still working to complete the phase 1 study. But interesting discussions, I'm sure, lie ahead.
Great. And a separate pushback, having spoken to yourselves as well as each of the companies in the TTR space already, has been around commercialization. Some of the existing players have argued that the cost of a gene editing approach would be prohibitive, given a one-time expense, and this in particularly the structure of payers and reimbursement in the U.S. Could you lay out how you see potential cost offsets in the existing therapies priced at between 1 to 2 Ferraris a year currently on a net basis, and the difference in the COGS and potential price flexibility that you have with an LNP approach versus the gene therapy comps, which require large quantities of manufactured virus?
Well, first of all, Manny, just to be clear, we don't have a viral approach here. So if people are invoking that as a base of comparison, that would be misguided. This is a manufacturing process that starts with essentially with small molecules or nucleotides. We have a phenomenal tech ops team who are charged with bringing efficient manufacturing procedures to not only our clinical trial material, but also to the marketplace when we get there. And I'm quite confident we will have a very, very competitive offering. I hear a lot about pricing. Since we've never offered an approach or a price, that is a straw man put up by people who do that at their own peril. And so I would say that we're going to be in an excellent position to bring not only medical value to patients, but I fully expect that we'll bring value to the healthcare system, and that's our objective here.
Thanks for clarifying that. I'll hop back into the other questions. Congrats again.
The next question comes from Lisa Baco from Evercore ISI. Please go ahead.
Hi, congratulations on the data, and thanks for taking the question. Can you maybe talk about, as you sort of envision the journey from here to sort of phase 3, what are kind of the next steps? And as you think about a pivotal design, can you maybe talk through sort of the duration, kind of what size? And I know it's still TBD, but, you must have some framework and sort of like guideposts on how you're thinking about that. And then also what kind of endpoints you're looking at. Thanks.
Thanks, Lisa. I'm going to ask David to give some general concepts that we're working through. David?
Yeah, good. Thank you, John. So the next step is really to get to the recommended dose for the phase 3 studies. So as you know, we're already at a point where we're getting a mean of 87% reduction. We have already started the dose escalation, the next cohort, so that you have a sense of where we're going in terms of where we want to be with dose. We do think we're fairly close, given that we're already have greater reduction than the existing agents, and we do think that there's a big advantage to that, as explained by Dr. Gilmore in his presentation. So once we do have that dose, based on all the data coming from our phase 1 study, we really want to be careful in that selection. We will go on to these more advanced pivotal studies. I should say we're already planning that. We will be discussing it with health authorities around the world, the design of that trial. I think you can get a sense of what it will be from some of the other studies that are already ongoing. We don't think they're very far off in the way they design the studies, but when we get closer, we will talk about more precisely what that design is.
And how do you think about duration? And then I just, that's a follow-up to that question. And then I just want to get a sense on sort of what incremental efficacy do you foresee between, let's say, somewhere in the 80% reduction in TTR versus if you kind of, migrate up into the 90s. Do you expect a material difference in terms of clinical outcomes, or how should we think about kind of as you have additional, reduction in TTR, how that might affect clinical outcomes as you go from this kind of 80 to maybe 90% range, and how we should think about, other compounds that are kind of in the 80% versus, inking something a little bit higher than that?
I think one thing to think about is what Dr. Gilmore showed is that for other diseases of amyloid, when you go from 90% reduction to complete remissions, you get a major improvement in survival in that case. And if you also think about another way, going from 80 to just 90% is already a 50% reduction. You've gotten rid of half of the residual protein. So that if the body's trying to get rid of the amyloid and there's half as much protein in the serum, that's why we think that's happening, as explained by Dr. Gilmore. So I think that's an important piece.
Okay, thanks. And then just on the, as you think about the duration, that was my follow-up question.
Yeah.
Thanks.
So in duration, understanding the mechanism of action, we are precisely targeting the TTR gene. That gene is no longer an effective gene. What we've shown preclinically, first of the non-human primates, that looking as long as you can look, beyond a year, you see no change in the reduction in TTR. In addition, we've done another experiment in mice that's shown in the New England Journal. And if you take out 2/3 of an edited mouse's liver, it will regenerate. And what we see is in the regeneration, the new cells are still edited. So the precursor cells are getting edited. And what we think that means, and that will be permanent. Of course, we need to show that. And we will be bringing you data later in the year. But that's what we expect based on the mechanism of action.
Okay, thank you.
The next question comes from Joon Lee from Truist. Please go ahead.
Hi, thanks for taking our questions and congrats on the impressive data. Have you harvested and sequenced different tissues, specifically the testes and the ovaries of non-human primate models that were dosed with the NTLA2001 equivalent for on and off target edits? And what will be the risk of transgenerational inheritance of these on or off target edits? And lastly, what are you doing prospectively to monitor for such outcome? What would the FDA want to see, and when can we expect the US IND? Thank you.
As part of the preclinical work, we do biodistribution studies, and what we see is that the LNPs go where you would expect them to go, which is primarily to liver and a few other related tissues at to very, very low levels. The risk of transgenerational passage we think is de minimis, and we have no evidence to indicate that that is anything other than a theoretical concern at this point. I guess, if we have offspring as a result of the study that, subsequently, I'm sure there'll be some interest in testing those patients, but the likelihood of finding something, I think, is de minimis.
The next question comes from Steve Seedhouse from Raymond James. Please go ahead.
Good morning. Congrats to everyone involved at Intellia and in the trial. I have two questions about NTLA2001 and two very quick and related ones about HAE and the guide RNA. First, as part of the TTR development program, any plans on enrolling switch patients from RNAi therapy?
David, do you want to?
Yeah, thanks. Yes, as part of in part 2, we do allow patients to switch from RNAi to our therapy.
Awesome, thank you. And Dr. Gilmore, you also showed the TTR reduction versus MNIS plus 7 dot plot for patisiran RNAi, but the same chart for antisense shows no correlation with TTR reduction despite having some patients that are like 80-90% reduced. And I've never found a good explanation for it. I'm just curious if you have any comment as to why there's no correlation for antisense or what the sources of uncertainty are in quantifying TTR reduction.
Julian, if you want to speak to that, feel free.
Yes, absolutely. So, thank you. Yeah, there's a very clear correlation, as I showed in every type of amyloidosis that's ever been treated, between the degree of knockdown and outcome. And I think the thing to say in ATTR amyloidosis is that the data are fairly immature. We've only been able to knock down TTR with gene silencers for about 5 or 6 years. And there's a correlation emerging, but at the same time, the rate at which a patient or a particular individual gets rid of amyloid is always slow, but it does vary a little bit. So it's not a linear correlation. So you need a good deal of data with a good deal of patients to really establish that correlation strongly, and I'm sure it will emerge as it already is doing so, but I'm sure it will become firmer and firmer as we get more experience.
This is very good. Okay, and for the company, the TTR knockdown that you're seeing, or reduction rather, that you're seeing so far in 6 patients, looks like it's tracking with your guide RNA adjusted PK/PD model versus the equipotent assumption, and you published both of those. So I'm curious for your HAE product, are you selecting dose levels that adjust for the sgRNA potency differences, and what is the potency difference for the HAE guide RNA in humans versus primates?
Thanks for the question, Steve. We're not going to go into relative potencies of guide versus NHP guides and different indications, other than to say the following. And I spoke to it earlier. We think we have very robust preclinical models. We feel very heartened now with the data we have that those models are very, very good guides to what to expect to see in the clinic. And we'll take all of that information, including preclinical work for HAE, as we figure out where we're going to start in that next study. And I hope to be able to report similarly robust findings, which we'll do when we have them.
Okay, terrific. Last question. I do appreciate it. Is it important in the TTR study to enroll cohort 4, even if you've optimized TTR reduction in cohort 3, just so you can get the LNP safety data in the event that HAE or some other indication down the road doesn't have, or 4 to 5-fold more potent guide RNA, or would you just dose the higher LNPs down the road if you need to and stop at cohort 3 here? Thank you.
Thank you for the question. What we really like where we are right now is that we're well into a therapeutic desired target space. There's no obligation to treat or to work through all 4 cohorts. There is some flexibility in the protocol, however, for us to explore intermediate doses should we desire to do that. That's something that we'll confer with our investigators and experts to think through what the merit of that actually is. Obviously, we want to be in a position to have a good dose selected as we expand the patient population in step 2. And we're looking for as much information as we can get to make the best possible decision. So lots of interesting assessments lie ahead. And as the information comes in, David and his team, I'm confident we'll make a good decision.
Thanks. Congratulations.
Thank you.
The next question comes from Mike King from H.C. Wainwright. Please go ahead.
Good morning, guys. Sorry, I lose my voice here a little bit. Thanks for taking my call, and let me add to the congratulations, especially by you guys hitting the marks so nicely on the initial dosing levels. A couple of science geek questions. I'm just curious, when you look at the supplemental data from the NEJ article, at the 0.3 mg per kg dose, you had first of all, you had 3 different genetic subtypes. Is there any correlation between the genetic subtype and the one patient who had the 96% response reduction in TTR?
The answer is no, not that we can draw.
Okay. And then further to that, the spread between the responses seem to be quite a bit larger. I don't know if that's just the play of small numbers or whatever the case may be, but in siRNA dogma, the higher the dose, the lower the variability. So I wonder if you wanted to maybe comment on that.
Well, we were pleased with how tight the data is and really gratified with that. And you're right. As you reach a maximal effect, one would expect that whatever variability there is to really start to collapse. Once you hit the maximum effect, that's 1/2 of the variability. And we're very, very close to that now. So I would expect with the ongoing work that that's the sort of observation we'll have, but that lies ahead. And when we have the data to look at, I'm sure we'll be sharing it.
Okay, super. And also wanted to talk maybe about the decay curves. It seems like they're sort of biphasic. I'm just wondering why, if you've looked into that, maybe from a preclinical standpoint of, there's some sort of biphasic effect going on, or, would we expect some more linear response rates as we get greater numbers?
Well, within a patient, there are different compartments. You have a protein that needs to be cleared. You have mRNA that needs to be cleared. And then at the beginning, there's the early effect on the gene itself. So there are compartments, but all that plays out very, very early on. And, the idea here is that once the gene is inactivated, the maximal effect is the effect, and that's invariant over the course of, we believe, the patient's life. So while the compartments are interesting, probably in the first few days, relative to how you think pharmacokinetically about a standard drug, it really doesn't apply thereafter.
Right. Okay. Well, thanks for indulging me on that. And then just finally, from a clinical standpoint, can you share with us your thoughts on the timing of when you might start looking at the wild-type TTR patient population? Do you want more, dosing and safety experience, or do you feel good enough with the data that you have now that the wild types might be on the docket fairly soon? Thank you.
David, how are you thinking about wild types?
Yeah, so we do expect those patients to have the same effect with the drug. So that's we think we'll know the right dose to go with. And as I sort of mentioned, we're already in discussions about how to expand the population to the patients with cardiomyopathy and wild-type TTR. So you'll be hearing more about that in the coming months.
Great. Thanks so much.
This concludes our question and answer session, as well as the conference. Thank you for attending.